Hey y'all, it's Ryan, and today I want to tell you what happens in your body focusing on your immune system when you have a short but stressful experience. The paper I'm talking about today reviewed and synthesized the results of 30 studies that used some kind of an acute stressful experience in the lab and then measured changes in inflammation from before the person was ever stressed out. From years of studying animals and humans, we know that our immune systems respond to short-term or acute stress. And as you know from the last video about inflammation, go check it out if you haven't already, it'll help some here. There are lots of markers of inflammation and immune function. So in the past, stress researchers have studied acute stress and a lot of these different markers, like the number or proportion of T and B cells, for example. But regardless of the specific inflammatory outcome, the goal of these studies is to understand better how psychosocial factors like stress or depression put people at greater risk for diseases across their lifespan. And the immune system is one key element in this understanding around stress and risk for disease. So naturally, we want to know associations at any one time point, so cross-sectional data, and over time, so longitudinal data. But we often also want to know how inflammation or immune function is impacted by stress in the hours immediately following that stress. So this is an important fine-grained kind of analysis, essentially of stress reactivity. We focus on stress reactivity assessed by cardiovascular or immune system changes because it seems like a promising explanation for at least part of why stress is associated with so many bad outcomes for us humans. This study asked 4 central questions, and I'll focus here on the first 2. So, which inflammatory markers are sensitive to acute psychological stress in humans? And secondly, what is the time course of these responses and how soon do they appear after that stress exposure? The authors here focused on cytokines, both pro and anti-inflammatory, so meaning either encouraging or discouraging inflammation, to see which changed following acute stress. They also looked at different ways that cytokines could have been studied, so either studying circulating or studying changes in cytokine production after these immune cells are stimulated with some kind of a toxin. Now you should check out this first video about inflammation for more about these different methods. Ultimately, though, there were only enough studies measuring these specific different markers to focus on 4 different cytokines: interleukin-6, tumor necrosis factor alpha, C-reactive protein, and interleukin-1 beta. So when focusing just on changes in circulating cytokines, there is a significant increase in IL-6 and IL-1 beta, with less robust evidence for a change in C-reactive protein. Here, TNF-alpha did not significantly increase over time. And there was a stronger effect for IL-6 when the researchers took blood samples for up to 2 hours, which means that the change in IL-6 might not be immediate, but slightly delayed as it increases in the 30 minutes to 2 hours after the stressor. So when immune cells were stimulated by a toxin to produce cytokines before and after a stressor, there's only a significant increase in IL-1 beta and none of the other markers. To sum it up quickly, we need a lot more research. The evidence here suggests that serum versus stimulated cytokines increase after acute stress, and that the timing of these measurements likely matters. There are lots of questions left though, and the paper does talk more about hypothesized mechanisms and factors like health status that might also affect who has a larger inflammatory response to acute stress. So make sure to check out this specific paper and let me know in the comments if you're interested in learning more about the details on this topic. Alright y'all, that's all for today. Thanks for stopping by and click like or subscribe for more videos like this.